So, and then we can come to today's speakers. So today we have two excellent ECI speakers. They will talk for about 20 minutes, and then there will be about 10 minutes for questions and discussions after each talk. So feel free to drop questions in the chat or raise your hand if you want to voice the question yourself. And later, after the at the end, we will also have some time for more questions if there are more that have not been answered yet. The recording will be probably posted at the Earnest YouTube channel later on, and you can then find the link on the Earnest website. Good. So I think that's all the important information for now. And then we come to the most important part, which are the speakers. And our first speaker today is Alessandro Nicoli from the Leibniz Institute for Food Systems at the Technical University of Munich. And Alessandro has graduated from Italy in pharmaceutical chemistry and technology in 2019. And he's now a PhD student in the group of Antonella Di Pizio and is working on modeling and simulating food-related GPCRs, including taste and odorant receptors, as well as neuropeptide receptors, which are also crucial for food intake and appetite regulation. And today he will focus on the molecular recognition of the neuropeptide Y receptor by the neuropeptide Y by the Y1 receptor, which aims to integrate various computational techniques to investigate the binding process of peptides into GPCRs. And with this, I'd like to hand over to Alessandro.
Yeah, thank you very much for your nice introduction. I will share now my slides. And please. Are you seeing the slide?
Yes.
Okay, perfect. Okay, we can start. Good afternoon to everybody. And today, as introduced, we will speak about the recognition by the Y1 receptor of the neuropeptide Y. The neuropeptide Y system consists of three peptide ligands, the neuropeptide Y, the peptide YY, and the pancreatic polypeptide. And also the humans express four functional GPCR, the Y1, 2, 4, and 5, but they are able to bind these three peptides with different specificity and affinity. We concentrate our talk in the MPY because it's the most abundant neurotransmitter that is released in the central and but also in the peripheral nervous system. It is involved in many physiological functions, for example, food intake, stress response, and anxiety, but also related, of course, to disease state, for example, obesity and mood disorder. But if you want to design a specific peptide, you want to know, for example, the molecular, how this peptide bind to the Y1 receptor, for example. And we decided to study and start our journey with the Y1 receptor because it's the most well characterized in terms of available structure. We have structure in the active state, structure in the inactive state, but we have also the peptide ligands in its abound state with conformation coming from the X-ray and the NMR. And our goal is to investigate how the MPY bound to the Y receptor, how we go from the unbound state to the final activation state of the Y1 receptor. We start with the first goal of our conformational analysis of the unbound state. We collected 20 structures from the PDB of the MPY, PYY, and pancreatic polypeptide. And here we can see on the left the reference, the bound state. And here we can see some example of the unbound state of MPY. Then we calculated the RMSD of the carbon alpha, and what we emerge also visually and from the RMSD, that we have an higher flexibility of the C-terminus and also of the N-terminal part of the peptide. But interesting, there are some already in the experimental structure peptides that are while nice RMSD, but they are able already to be in a conformation like to be as the bound state. But we have also other, for example, the 1RON, that is totally completely different from the unbound state. Then we decided to go for the first molecular dynamic simulation, where we decided to simulate the peptides, the most diverse in water. And what we can see that after a few nanoseconds, already the alpha helix part already sample the bound state. And then also after some 100 nanoseconds, the N-terminus part refold back to form a stable conformation already starting from the most diverse MPY from the experimental state. Then we also performed the molecular dynamics of the bound state in water, and during the trajectory, it was really stable, meaning that this conformation is one of the most stable and can be sampled with the molecular dynamics. What emerged that as in the experimental structure, the higher flexibility is for the C-terminal part. But what driven the refold back of the N-terminus is driven by hydrophobic interaction of this unstructured part of the first 13 residues, but importantly, they are able to driven this conformation that is prone to the binding to the GPCR. Then we move to the receptor side, and what we can learn. Here we can see the full active state. If we superimpose the receptor in the inactive state, we see that we have also a big conformational changes of the extracellular loop 2 part, but also of the extracellular loop 3 part. We then decide to collect and then run a new simulation to increase our sampling of the extracellular loop part. And we have a simulation from the GPCRMD database of the APO, HOLO, and the inactive state, but also we run new simulation of the APO and HOLO state. We run using ACMD3 molecular dynamics engine with the force field CHAM36 and using membrane POPC, TIP3P water model, and chlorine and sodium ions. Then to map and to see this movement along all the trajectory, we decided two collective variables. In the y-axis, we will have the distance of the center of mass of atoms of the extracellular loop 2, and we will have atoms of the extracellular loop 3. And then instead on the x-axis, we will have here a representation of the movement and the RMSD of the carbon alpha of the binding site that comprise all the extracellular loop 2 and 3, and all our reference will be the active state. Then we will have in our plot the area where the conformation are in the active-like, and also where the conformation are in the inactive-like. If we project already just the experimental structure of the receptor, we can already see that the two inactive state have a similar aperture of the extracellular loop 2 and 3 distance, but they have already a different displacement of the binding site residues. Then we analyze all the simulation that we have. We will have on the top the two simulation. In dark green, we have the HOLO state. Here we have the active state without the ligands. And here in orange and shades of orange, we have the inactive state. Here we have bound to antagonist, and here we have the complete apostate trajectories. What we can see nicely that the ligands can be the antagonist of the peptides, stabilize certain conformation here on the right of the plot, but also interestingly, when the receptor is in the apo form in the inactive state, is able to open in a similar fashion of the active-like. And it looks like to be an intrinsic dynamic features of the receptor to opening, and vice versa, some of the apostate in the active-like is able also to reclose back. We can also extract some of the structure of the apostate. Here we can see the superimposition of two frames. In green, we have the active state. And we can see that the extracellular loop part already fold, and also nicely, the extracellular loop 2 fold this small alpha helix in a similar way to the active state like. Then, if we look on the recognition of the peptide to the Y1 receptor, what we can see that the most of the receptor engage interaction with all the extracellular loop part and protrude the C-terminus part inside the orthosteric site. We have that the N-terminal part make polar interaction with the extracellular loop 2 and the apical part of the transmembrane 5. We have that all the alpha helix make hydrophobic, but also a net of polars and salt bridge with the extracellular loop 3 and the N-terminus. And also the C-terminus point with a lot of 2 arginines and the last residues tyrosine with the interesting polar interaction. And just to remind that the C-terminus of the NPYs is also amidated, is important for the activity. But what we don't know is how the peptides bound and arrive inside orthosteric site. We then decided to perform a series of Walker supervised molecular dynamics, and we perform 5 replicas of the active state, and we perform 3 replicas of the best active-like conformation that we extract with our analysis. Here we can see one simulation of the entrance of the peptide, and we can see that the peptides start to bind close by, and the first anchor point are the residues at the D32 and the residues 10.4 at the extracellular, at the apical part of the transmembrane 2. And then if we continue our simulation, we can see that the C-terminus start to engage with the 3.32 and start to bring the C-terminus part inside the binding site. Then we merge all the trajectory that we had of the active state and we can finally have which are the residues that are important for the first part as an anchor point, these 2 aspartate, but which are also the residues that are crucial after the first initial anchor point. And then what was nicely that starting from the full active state, but also from the inactive state, we were able to sample the entrance of the peptide. Then, with the MGBSA analysis, we map the landscape energetic of the entrance of the peptides. And with this analysis, we can also nicely study which are the metastable sites that bring the peptides inside the G protein. Another interesting came out that it came from the inactive state conformation. Here we can see the starting plot. We have in green 2-phenylalanine that when it is in the active, they protrude towards the active state. But when we start our simulation, they were in an active state like conformation of 2 residues. But when we have the bind of the peptide, we see that the peptide induced the conformation of these 2 residues to be in an active like when we have an opening, but then the peptide, when bounds already in the starting from the inactive state, is able to bring an induced fit form that bring the 2 residues to have an active state like conformation. Then, if we have a look at the final snapshot of our replicas of the SWAMD, we can see that we can easily, nicely sample on the left the conformation of the active. But what was difficult to achieve was a complete superimposition or sampling of the C-terminus part. And for starting from the inactive since it has to open more, we do not have a full completely protrude of the C-terminus inside the orthosteric side. We then have a look also on the simulation when the peptides is already bound MD simulation of the active Y1 receptor bound to the MPY. And what we can clearly see is that the C-terminus is really stable after 500 nanoseconds. If we can see here the MSF plot, the RMSF is a value that gives you how much flexible is a certain area of a protein or a peptide, we can see that the C-terminus it's really stable. And also here on the right plot, we have the RMSD of the side chain, and they do not go beyond 2 angstrom, and it's pretty stable during the simulation, meaning that this conformation of the bound state is one of the most energetic stable that we can have. We decide to have a more focused analysis but focus on the C-terminus using a molecular peptide docking. We took the unbound peptide that we have. We apply a cutoff of 5 angstrom of the best frames with respect to the active conformation. And we using Class Pro, and we can see here on the right the results of the best models that came out. And we can see that we have some already with the docking that we can sample for the RMSD of the whole carbon alpha already too. But also we can have a focus C-terminus RMSD with a really low, but also have a sampling of already using Class Pro, starting from a different conformation of the side chain of the peptide. We then perform a molecular dynamic simulation also of the our best docking pose, and what you can see as in a similar fashion to the crystallographic pose, we can see that the C-terminus, when he founds this conformation, it stays stable also for all the trajectory. And here we can see the same plot that we see before about the RMSD, and we have a similar behavior. And with the docking, we can optimize the final state also that we can sample during the entrance. When we come into our first slide that we wanted to analyze the molecular recognition, what we learn from our journey. We learn that from our analysis, the mechanism is driven by a conformational selection, because the MPY, the peptide ligands, can sample already in the NMR structure, but also in the MD simulation, the bound state. But also that the inactive state conformation is able to open the SL2 and the SL3 in a similar way to the bound state. From the SWMD and the peptide entrance, we came with the important residues that are important for the as anchor points. We can have which are the metastable point along all the pathway, but also we were able with the inactive state conformation to see also a rearrangement of the residues from the inactive to the active-like states. And all those residues, interesting, are also in agreement with mutagenesis data. And then that the C-terminus probably is the key step for the activation. And we propose also the focus docking also of the C-terminus part. I want to thank all my group, my supervisor Antonella Di Pizio, of course Giuseppe De Ganuti for the help with the molecular supervisor molecular dynamics, and also Ernest to provide the all the training school and also this beautiful GPCR. All of you for your attention. I'm happy to take all the questions that you want, and you can reach me in Twitter also too. Thank you very much for the attention.
Thank you very much for this great presentation. So, yes, please post questions to the chat or raise your hand if you would ask something. There are already 2 questions in the chat, so maybe we'll take these ones first. So the first question is about the terminal patches that you used for the simulations, or whether you used the ends uncapped.
We used for the N-terminal part, we need the first residues, we maintain it charged. Instead, for the C-terminus part, has been amidated with the C2. We use the charm force field, and the patches were the C2, CT2, and we use the. Of course, because who knows, of course, the MPY, but in literature, when this amidated C-terminus part is not present, the peptide is not active.
Okay, and then the second question was about the loops and whether they actually need to open for the ligand to get into the binding site. You're muted again.
Sorry, could you repeat the question? Sorry, because I do not hear it.
It's also in the chat if you want to read it. But so it's about the whether you think the loop actually needs to open for the ligand to enter.
What we're thinking, basically that we have a first aperture, basically already in the simulation, the ACL2 and the ACL3 movement can already open. But of course, not in all the residues can be already in a similar, of course, way of the full active state. We think that is a two key step. We have first, that in general, we have the peptide already can be in a bound state and the receptor can be open and can close. But at the same time, when the peptide entrance inside the binding site, then there is, of course, an induced fit, but locally. We think that there is a conformational selection already, but when the peptides is inserted, it is already inside the binding site, there is an induced fit, but induced from the peptide. And of course, then we have the full active, but this one we have not explored from this inactive like to the full active state. Is work to do now. But thank you for the question.
Okay, and then we have another question. Which is about the Walker supervised NDs. Whether you could tell a bit more about it. So the protocol and how they are different from normal replicas that start from different initial configurations, if there is a difference.
Okay, we have basically we have in chat for sure Giuseppe De Ganuti, that is the developer of this multiple workers from D. I will try to explain it, but of course I will also happy to hand over the more question to Giuseppe, and I will try to share a small slide about it. And I think, give me a second, that I will try to share here. I think. If you give me a second. Okay, give me a second that I go. I have a slide ready for this. Okay, perfect. I think now you should see my normal slide. You should see the slides, and basically, okay, yeah. Basically, I will start from the classical supervised molecular dynamics that was developed in Padua by the group of Stefano Moro. And basically, what the original algorithm does is basically it sample on the way you start from a distant point. You have, for example, the center of mass of your binding site, the center of mass of your peptides. And you start from an initial configuration, you perform a small series of classical molecular dynamics of, for example, 600 picoseconds. Then you took all the points along the trajectory, you fitted. If the algorithm works like that, if we have let's say, the peptides or the ligands move close to the bind site, then it will forward to the next step and it continue. Otherwise, it will start with a randomized event. And this one, it goes till when we have a distance of center of mass between the peptide or the ligands and let's say our target. When it's minus 5 angstrom, it starts with a classical molecular dynamics. This one was the original code. Then, let's see in the chat if we have okay, and there is Giuseppe that he can pick up the question and now explain the protocol in a better way.
Hi, thanks, Ale.
No, thank you to Giuseppe.
No, okay, the one you showed, basically, yes, it's the original implementation of the supervised MD, and it was mostly applied to the study of binding events, ligand binding events, and then peptide protein events and RNA target events. With the multiple worker SWMD, we try to extend the applicability domain, changing a bit the approach. So instead of performing simulation in series, you do parallel simulations, and you can supervise the distance between two molecular objects, but also the MSD and contacts, hydrogen bonds. The goal was to try to study also conformational changes and those kind of different GPCR important phenomenon. So yeah, I really, if you want to extend the reading about that, Alessandro, you put something in one slide. I think the link.
Yeah,
to
I can share the link of the preprint that you can have the preprint of the original code developed by Giuseppe.
Take-home message is just that the supervised MD is a tool for simulating protein transition, structural transitions. Okay, that's the main goal. Thank you.
Thank you very much.
Okay.
Sorry, yes.
Yeah, so we can go on with our second speaker of today. She's Katie Sharrox. Katie graduated from the University of Sheffield in 2020 in biochemistry and genetics. She's currently a PhD student, her final year of a PhD in the Imperial College London in the research group of Professor Eileen Anialoglu and Professor Bernadette Byrne. Her PhD is focused on D2 dopamine receptor oligomers. Using mutagenesis-based approach, the aim of her PhD project is to identify receptor mutants which can increase dimer stability and develop pathways for high-resolution structural analysis. Some receptor mutants have been identified which have interesting functional effects, and so this talk of today outlines the progress towards obtaining an optimally stable D2 homomer and the impact of these mutations on functional activity. Please, Katie.
Thank you for the introduction. I'll just share my screens. Hopefully you can see my PowerPoint. Yeah, okay. So as I was introduced as, I'm going to present some of the work that I've been doing during my PhD and presenting some work on a mutagenesis-based approach to stabilizing D2 dopamine receptor homomers. So just to introduce the D2 dopamine receptor for those of you who are not familiar with it, it's a class A G protein coupled receptor that can exist as a monomer, but can also form homodimers or higher order structures, and is known to form heteromers with other GPCRs, including the A2A receptor. And the D2 receptor normally binds to G-alpha I, so it inhibits adenyl cyclase and causes a decrease in cyclic AMP production. However, it is also known to signal through beta-arrestin 2 signaling pathways, including through activating the MAP kinase pathway and inhibiting the AKT pathway. So the reason these receptors are particularly important is that they're implicated in a number of neurological diseases, including Parkinson's disease, schizophrenia, and bipolar disorder. And a few different receptor states are associated with different diseases. So, for example, an increase in D2 homodimers has been associated with schizophrenia, and D2A2A heteromers have also been associated with Parkinson's disease. So we have high-resolution structures for monomeric D2 receptor and A2A receptor, but what we don't really understand is the molecular interactions between receptor proteinuria and dimers. So that's where my PhD project comes in. We really want to understand how these receptors form dimers and what the precise interface is between them. So we decided to use a mutagenesis-based approach to try to stabilize D2 homodimers for high-resolution structural analysis, primarily by cryo-electron microscopy. During this presentation, I'm going to highlight some of the first two aims of my PhD project, which is to functionally characterize some D2 receptor mutants that I've made and assess the impact of these mutations on the stability of D2 homodimers or oligomers. So the story of my PhD actually starts with the A2A-D2 heteromer. So originally my PhD was going to be looking at the heteromer, and our collaborator, Francesca Finelli at the University of Modena, did some molecular docking simulations to come up with a predicted heterodimer model. And this model that she came up with predicted a TM1, TM2 interface. And she's identified residues that lie at the interface that, when mutated to cysteine, are in close enough proximity that they have the potential to form disulfide bonds between the individual protomers. So the first step that we did to look at the impact of these mutations on stability of dimerization was to do a Western blot under reducing and non-reducing conditions. So I've designed receptor constructs that have an M-terminal flag, so we can recognize the receptor using an M1 anti-flag antibody. And what we actually found, which was unexpected, was that the D2 receptor mutant V96C actually increases the homodimer stability rather than the heteromer. So from now on, we switched to looking at the homodimer to investigate this interesting finding further. So we saw that under reducing conditions, the relative levels of D2 protein in the dimeric form compared to the monomeric form is higher in the V96C mutant compared to wild type. And to provide further evidence for this, we did some Brett saturation assays. So I won't introduce this too much to this audience, but this is a biophysical assay that allows us to look at protein-protein interactions. So in this case, I had an R-Luc protein fused to the C-terminus and a YFP to look at dimerization. So when dimerization occurs, you get resonance energy transfer between the R-Luc and the YFP, which can be detected at 535 nanometer emission. So by transfecting HEC293 cells with either wild type receptor or V96C receptor, we transfected increasing concentrations of the YFP tagged receptor and constant amounts of the R-Luc receptor. And we obtained a saturation curve, which indicates dimerization. And we can quantify the Brett max levels and the Brett 50 levels from these curves, and we observed that there was an increase in Brett max with V96C compared to the wild type, which may indicate an increased protemer proximity. So given this interesting finding that we saw with the D2 homodimer, we went back to Francesca and she did a molecular docking simulation of the D2 homodimer this time, and she identified further mutations that she thought would increase the stability of the D2 homodimer specifically. Again, we did a Western blot to look at the impact these mutations had on dimer stability. And we found that the double mutants, the V96S, V97C mutant under reducing conditions, seems to have much more of the dimeric form of the protein relative to monomer levels when compared to wild type. And this effect seems to be independent of disulfide bond formation, as you don't see the complete destruction of the oligomeric form of the protein when we do this under reducing conditions. So to assess the other aim of my project, to look at the effect of D2 mutations on the function of the receptor, we first looked at GI signaling. So to do this, we did a cyclic AMP assay. So in HEC293 cells transfected with the wild type receptor or the mutant receptor, we stimulated the cells with forskolin, which is a direct activator adenyl cyclase, and treated the cells with increasing concentrations of quinpirole, which is a D2 selective agonist. And from these dose response curves, we saw no observable difference in the IC50 values. So mutating the receptor doesn't seem to affect the potency of quinpirole to activate GI signaling. However, one thing I did observe was that at the dose which gave the maximal GI response, the levels of V96C and the double mutant showed less GI activation than the wild type. So to look at the other side of the signaling, we looked at beta-arrestin 2 recruitment to the receptor, again using a Brett assay. However, this time the R-Luc tagged receptor was used, but the YFP tagged protein was beta-arrestin in this case. So an increase in Brett ratio indicates that beta-arrestin 2 was being recruited to the receptor. So we were able to do Brett assays in real time to visualize beta-arrestin 2 recruitment and analyze the kinetics of this recruitment. So when we add quinpirole, you have an increase in the Brett ratio, which indicates an increase in recruitment of beta-arrestin 2. And when we take the basal and quinpirole values away from each other, we obtain a ligand-induced Brett ratio. And we can plot this on a curve, and the waveform produced can be quantified to analyze the kinetics of this recruitment. So we found that V96C seems to recruit beta-arrestin about 2 times as fast compared to the wild type receptor. And we analyzed this for the other mutants, the V96S and the double mutant. And this didn't seem to affect the kinetics of arrestin recruitment too much. However, it seemed to be that the V96S and more so the double mutant had a higher Brett ratio under basal conditions, which may suggest that these mutants are recruiting beta-arrestin 2 more readily under basal conditions. And the V97C and the Y93C mutants seem to have very similar kinetic profiles to the wild type receptor. So these are very similar assays that we did. Again, Brett assays to assess beta-arrestin 2 recruitment, but this time a dose response, so increasing concentrations of quinpirole were used, and we quantified the EC50s of these curves. So with the V96C mutant and the V96S mutant, you see a left shift in the curve, indicating an increase in potency of quinpirole to induce beta-arrestin 2 recruitment. However, there was no change with the double mutant or the V97C or the Y93C mutant. And with the V96C mutant, I see an increase in ligand-induced Brett ratios, which again indicates that this mutant recruits beta-arrestin 2 more readily in the presence of agonists. And I also saw that under basal conditions, V96C seems to have a higher basal Brett ratio. So overall, this may indicate that the V96S and the double mutant seem to recruit beta-arrestin 2 more readily under basal conditions, but less so under ligand-induced conditions. As we saw this altered beta-arrestin 2 recruitment, we wanted to see whether this affects downstream signaling. So in HEC293 cells transfected with wild type or V96C and stimulated with quinpirole for different treatment times, At 5 minutes quinpiral treatment, we saw a significant increase in phospho ERK activation, so an increase in the MAP kinase activation pathway at the 5 minute time point compared to wild type. And this seems to be dependent upon beta-arrestin 1,2, as in knockout cells. This observed increase in phospho ERK seems to be abolished and is very similar levels to basal. When we looked at this in cells transfected with V96S or the double mutant, they seem to actually have a lower level of MAP kinase activation, so lower levels of phospho ERK protein compared to wild type. However, part of this may be due to the increased basal beta-arrestin to your recruitment, so you see less of this ligand-induced response. It could also be the V96S, V97C double mutant that I'm seeing higher levels of basal MAP kinase activation. So this is again reducing the ligand-induced responses. And finally, I just want to introduce you for some stuff that I've been doing more recently. So I've been doing some super resolution microscopy techniques to visualize single GPCR molecules. So this is a super resolution microscopy technique, PD-PALM, which allows 20 nanometer resolution and visualization of individual GPCR complexes, so monomers, dimers, or oligomers. So this process, the PD dyes that we use are photoactivatable. So when you expose these dyes to a laser, this causes uncaging, exciting, and eventually bleaching of the dye. And these dyes are specific to the receptor. So what we get is these nice images of flashes or blinks, and these individual dots are individual GPCR molecules. So through processing this through a QuickPalm software, we can localize these individual GPCR molecules to specific XY coordinates. And after filtering and putting it through an interpreter, we use Gettys-Franklin neighborhood analysis to identify whether an individual molecule exists as a monomer or a dimer or a higher order structure. And it produces this heat map here that we see on the right. And quantification of this under basal conditions, if we take the individual populations expressed as a percentage of total receptors, we see this significant decrease in the number of receptors that exist in the monomeric form of the receptor, with the double mutant and to some extent the V96S mutant. And when you express this as non-associated or associated, so monomeric or oligomeric form of the receptor, you again see this decrease in the non-associated and increase in the associated form of the receptor with the double mutant compared to the wild type. So this preliminary data under basal conditions may suggest that this mutation shows preference to associate in oligomeric forms rather than monomeric forms. So to briefly summarize, I've created this figure to show some of the most interesting findings of the receptors that I looked at. So overall, the mutants that I looked at show different effects for different types of functional or stability of the dimer. But overall, during basal conditions, all of these mutants, the V96S, the V96C, and the double mutant, show increase in oligomerization under basal conditions. V96S and the double mutant show an increase in basal beta-arrestin 2 recruitment and potentially also basal MAP kinase activation. And under ligand-induced conditions, so in the presence of agonist, V96C and V96S see an increase in ligand-induced beta-arrestin 2 recruitment. And with V96C, this is translated to more MAP kinase activation after 5 minutes quinpiral treatment. And V96S and the double mutant see a decrease in GI signaling. And V96S shows a decrease in ligand-induced MAP kinase activation. So overall, the take-home messages, I know I've shown you a lot of different effects of different mutants, but overall, we have identified D2 mutants that exhibit altered functional characteristics, and some of these may show potential bias towards beta-arrestin 2 signaling pathways. And V96S, V97C, a double mutant, is a particularly promising candidate, we think, to form the basis of future work towards using this in high-resolution structural analysis. It's possible that D2 dimers may favor a conformation that promotes beta-arrestin 2 recruitment from this data, and this finding could be particularly important for therapeutic drug targets, and maybe designing disruptors that disrupt the D2 homodimer to produce more specific drugs. And with that, I'd like to acknowledge both of my lab groups, and particularly the Hanyaloglu lab group and my master's student, Yue, who produced some of the BRET data that you saw. And thank you to everyone for listening. And I'll take any questions now.
Thank you so much, Katie. Very nice presentation. Congratulations. So we have a question from the chat. Does adding the agonist actually change dimerization dynamics? Does it do it differently from mutants? Also, maybe it will be interesting to include FAB30 or other arrestin conformation-specific nanobodies in your experiments to actually check if promoting dimerization changes the way arrestin binds to the receptor.
Yeah, so that's a very good question. What we're looking to do hopefully soon is to look at dual-colored palm to look at arrestin recruitment to the receptor. So we'll be able to visualize using palm whether the specific GPCR molecule is in a dimeric form, and does it have, is it more likely to associate with arrestin? Also doing molecular dynamic simulations would be interesting to see whether making these mutations affects how it associates with arrestin. But as I said, we're currently hypothesizing that these mutations increase dimerization, and that also may increase the likelihood that it recruits beta-arrestin or how readily it recruits beta-arrestin. But at the moment, we don't have that data directly linking the two. But yeah, it's a very good question and one we hope to address.
Great. Actually, I have a question for you. I was wondering, because did you think to test biased ligands of the D2 dopamine receptor and see if the dimerization alters their biased behavior?
Yeah, that would be really interesting to look at. It's something we did consider, and maybe helpful for when we want to try and express and purify and do the structural analysis work to further try and bias it towards beta-arrestin if we think that's causing the receptor to be more likely to dimerize. Yeah.
Thank you. So, is there any more question from the audience?
Maybe I can ask one. Thank you, Katie, for a very nice talk. I was wondering if, because if I understood it correctly from the docking poses, these point mutations that you come up with are in the TM in the helix 8, if I'm not wrong.
Most of the mutations are on the extracellular loop 2 or helix, transmembrane helix 2 of the receptor.
So none of them were in helix 8, because at least what I understood from the pose that you showed was that they were in there. And yeah, just if it's not, then ignore, because my question was that maybe you are playing with the phosphorylation sites in the helix 8, and then it may explain why you're getting higher beta arrest in recruitment.
Yeah, the mutations, they're just on the edge of the transmembrane helix, so they're more on the extracellular side rather than helix 8. But yeah, that's a good point, yeah.
Thank you.
Maybe I can quickly follow up on this question, kind of, because what I was wondering is whether your assumption is, or your model proposes, whatever, that these mutated residues are involved in direct interactions, either with each other or like with another part of the receptor, or whether it could also be some other effect that like changes maybe the conformation and therefore they can dimerize better. Do you have any like ideas, hypotheses, models on that?
Yeah, so yeah, it's a good question. So most of the docking simulations were done in active state structures. So what we don't really have a lot of data for at the moment, particularly because all of my palm data so far is during basal conditions rather than in the presence of agonists. It'd be really interesting to see what effect adding agonists has on either the dimerization or how it affects the molecular docking simulations that Francesca did. But yeah, it'd be interesting to see if different conformational states have different effects.
Yes.
Good.
Sorry. Because also I was wondering, one of the residues is mutated to a cysteine, right? Could something happen, like are they located in a way that it could be actually like linked by a disulfide bridge? So are these dimers dynamic or do they stick?
Yeah, that's possible. So the cysteine mutations, the reason we chose those mutations specifically was that they were predicted to form a disulfide bond with the other protemer. So what we expect and what we want to happen is for that disulfide bond to form to try and stabilize the dimer. But how that affects how well the receptor activates or how well it changes conformationally is something that we would probably need to look at a bit further. Yeah, it's a good point. So we want the cross-links and disulfide bridges for the purpose of the stabilizing the dimer and for the structural analysis, but what effect that has functionally may be interesting to look at, but may also have adverse side effects in terms of signaling.
Thank you.
Okay.
So, sorry, I think I will stop the recording now. So if anyone was too shy to speak while the recording is running, you will still have the chance now.